Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Sam Moulton from the Business Review, and I will be your host. It's our pleasure to have Ben Research with us today, who will be discussing the innovative PAT integrated bioprocess environment. For more than 40 years, Ben Research has worked with clients to create value by advancing new medicines that improve human health and to solve the most difficult scientific and technical problems. This success is based on the company's ability to develop, advance, and commercialize pharmaceutical technologies which grow from a solid base of scientific and engineering fundamental understanding. Ben Research, a division of capsule dosage form solutions, solves customers' most pressing product development challenges, including biotherapeutic formulation and processing. High-quality science and engineering is core to their offering at each stage of the product development cycle. Today's guest speakers are Jeffrey Bright, PhD. Jeffrey Bright is a director at Ben Research Inc., where he works in the fields of biotherapeutic drug development, inhalation drug development, and formulation technologies. He's worked at Ben Research for 7 years. Dr. Bright earned his PhD in pharmacology from the University of South Alabama while working in the Center for Lung Biology. His doctoral work focused on defining genetic and molecular signaling processes involved in pulmonary disease. Dr. Bright performed his postdoctoral work at the Roche Pharmaceuticals in Palo Alto, California, where he researched the genetics of complex disease states. And the other speaker is Brendan Downey, Technical Group Leader. He's a research chemical engineer and technical group leader at Ben Research, where he works on new technology development programs. His primary areas of expertise and responsibility include the development, scale-up, and integration of new manufacturing technologies, the biotherapeutic production processes, with the aim of obtaining better cell and product-based process guidance. He earned his BS in chemical engineering from the University of Colorado, Boulder. I'd like to welcome you to our platform On24. You'll notice that it's browser-based, so if you disconnect at any point, you can just click on the link that you received via email join. In order to ask questions, you can send these in via the Q&A widget, which is at the bottom of your screen, or just type them into the questions box, which is at the top left-hand corner. Try and allocate around 10 or 15 minutes at the end of the session to address any questions or thoughts you may have. If you click on the survey widget, there are a few questions in there for you to answer in order to derive feedback. You can do this during or immediately after the webinar is finished. If you click on the green resources widget, you'll find some links from our speakers to view. And if you require help at any point, just click on the yellow help widget or send in a question to me. You can move and resize and maximize any of the windows in front of you to get a better view of the slides. But now, without further ado, please allow me to welcome Dr. Jeff Bright.
Great. Thank you, Sam, very much for the introduction. So the title of today's webinar is Integrated Bioprocessing Environment for Understanding, Automating, and Controlling Protein Quality Attributes. And the goal of this webinar is to present work we're performing in the area of biologics manufacturing, specifically the integration of bioreactor sampling technologies with PAT tools and process optimization around critical quality attributes of therapeutic proteins. As stated in the title, we're in the process of tying these technologies together and using them to develop a control platform focused on optimizing protein CQAs. During this webinar, we'll be presenting a number of technologies that we're using and presenting a case study focusing on galactosylation. Over the last few years, we've presented our work in a number of places, including posters, talks, and webinars. This presentation is essentially an update on activities at Ben Research in this space. It highlights sort of where we've been, where we're going, and what our current thoughts are around product and process optimization. We think that with the correct tools, concepts, and data application, we can really leverage these areas to make strides in this space of process optimization. That's more or less what we're discussing today. So as Sam stated earlier, Ben Research has been around for more than 40 years. We've been operating in the bioprocessing space for more than 8 years. Ben Research is a division of Capsugel. As you can see in the bottom right-hand side of the slide, we are within the dosage form solutions umbrella of Capsugel. along with Capsugel Emerging Technologies and NCAP. At Ben Research, we offer a number of technologies around formulation and delivery, and today we'll be discussing in particular bioprocessing technologies available at Ben Research for our clients to work on and use. So working with Capsugel and Ben Research essentially brings to bear an access to a breadth of technologies, including bioavailability enhancement, such as spray-dried dispersions or hot melt extrusion, as well as modified and targeted release, such as multi-particulates, matrix, and osmotic approaches, for instance, as well as specialized applications such as inhalation and abuse deterrence, as well as taste masking and specialized manufacturing. On the far right-hand side of this slide, we see biotherapeutics. Under biotherapeutics, you can see guidance tools for bioprocess optimization, which is what we're discussing today, as well as biotherapeutic or protein formulation and development, including vaccines, peptides, millimolecules, and large molecules. So essentially, we've generated here at Ben Research these bioprocessing optimization areas by leveraging our core engineering, chemistry, and biology capabilities. This is an area clients have asked us to get involved in. We've been posed problem statements, and we've provided solutions as a result of our internal know-how and capabilities. Some of what you'll be seeing during this webinar is us using these capabilities to optimize manufacturing processes. Protein-based drugs are increasingly becoming important in treating disease. In order to facilitate the growth of this field, our development work is really focusing on delivering technologies around formulation and manufacturing and identifying solutions for our clients' problem statements. We are currently, as listed in Biotherapeutics, we're currently working in the area of formulation because it leverages existing know-how and capabilities, specifically excipient selection and spray drying. In particular, I'll be talking next week at the AAPS National Biotech Conference on protein formulation. Let me know if you'd like to talk then about that topic in particular. But for today, we'll really be talking about bioprocess optimization. This is an outline for what we'll be discussing, reviewing today, myself and Brandon. The work we're showing today has, in particular, one defined goal, but there are numerous goals internal into this. The goal is to develop an automated methodology for generating model-based process understanding and prediction of culture behavior. What that means is we're essentially leveraging internal hardware, software technologies, generating large data sets, reducing those data sets, and then understanding our system in a way that enables us to define processes, new processes and optimizing processes, and making predictive models of those processes. So from an outline perspective, the first thing we'll be talking about there, shown in blue, is an overview of our methodology. Essentially, we'll be discussing concepts that we're utilizing in this space. There'll be an introduction to areas such as system identification, process control. We'll be mentioning briefly hardware and software and big data and our motivations for utilizing those sort of technologies and that know-how. The next bullet point down is technologies developed to enable methodologies around process optimization. And we'll be discussing hardware, integrated lab environment, software, and once again, how we're handling large data sets and curation. Then Brandon will be discussing a case study wherein we leverage these capabilities to really begin doing a deep dive on protein galactosylation, understanding sort of first-order input-output relationships, looking at dynamics and steady-state galactosylation responses as a function of galactose. And then at the end of this presentation, we'll have a wrap-up and a Q&A session. So Brandon, I will hand over to you next few slides.
Cool, thanks, Jeff. So as shown on this slide, in many cases during the development pipeline of a biologic drug candidate, process development ends up on the critical path. So the sort of representation of a more or less traditional biotherapeutic development pipeline is shown on the top half of this graph. And there's really kind of two places where process development is really critical toward advancing a biologic drug. In the early phases, the goal is often to achieve just high enough titers in bioreactors to be able to supply material for tox and early phase supplies. So this usually requires its own set of bioreactor experiments to accomplish. If we move on down later into the development pipeline, substantial effort is then typically invested kind of prior to phase 3 to increase process robustness, particularly with respect to being able to reproduce product quality attributes. So in early phase development, since the effort is really often invested in more in achieving practical titers that can be used to manufacture tox supplies, the emphasis in this phase is often focused on really being fast and cheap, so that a quick early phase clinical readout can be achieved. And so understandably, kind of in these early phases, investment in process analytical technologies, automation, or in-depth process platform understanding it is typically not made at this early stage. So this understandably creates some challenges. One, this lack of investment can lead to a less of an understanding of the process platform as a whole, and that therefore leads to much repeated effort from molecule to molecule. Secondly, this often leads to substantial late phase process development effort being needed. And this is because the data that's required for the development of a scale-up and control strategy for each molecule that's in the pipeline for each candidate process has often taken a backseat to just kind of getting that early quick win with early phase supplies, and so therefore those in-depth high-quality data sets have to be regenerated later in the development. So as a result of those challenges, there ends up being a practical limit to how many molecules can be delivered through a process development organization. And this forces us to choose which molecules to advance before we have any clinical data to make a data-driven decision about which molecules to advance in the first place. So it's a situation that maybe we don't want to be in. So today we're going to present work that's ongoing at Ben Research to develop an integrated process development platform that we think can enable more candidates to make it to the clinic by noticeably shortening these development timelines and therefore lowering the process development effort that's required for each candidate to go through the pipeline. So in this integrated process development platform, we attempt to leverage automation as well as predictive modeling approaches to increase process platform understanding. So we see an opportunity here to apply these approaches in such a way so that we can streamline early phase development by automating experimental design and the collection of data from those experiments. And we see an opportunity to minimize late phase development by leveraging the data and platform understanding that we collect in early phase, essentially by collecting higher quality data sets in early phase development using an automated platform. So in this platform, candidate processes are introduced to the integrated process development lab, where both the experimental execution and the collection of data from those experiments are automated. And this obviously substantially lowers the amount of effort that's required to collect the high quality data sets that we need in early phase process development. Those experiments and the resulting data that are collected in this environment result in high quality data sets that we can then use to create data-driven predictive models. And those are models that are generated from the behavior of the process itself in the early phase. This predictive modeling approach allows the use of high-quality data to generate process understanding, which can be applied across multiple molecules, multiple processes. So this predictive modeling approach also allows the nomination of a control strategy for each process very early on in the development pipeline. So it also makes it so that the early phase data can easily be applied to process robustness studies later in development if required. So the goal of the platform that we're presenting here is to develop an automated methodology for generating model-based process understanding and prediction of culture behavior. Today we're going to go through some of the tools that we've developed to enable this approach in our shop here at Ben Research. So specifically, we'll show how experiments can be automated to yield the high-quality data sets that are required to generate predictive models and ultimately use that for process control. We're going to show how we utilize automated bioreactor sampling technology, such as MASS, to collect high-quality data sets from these automated experiments. We're going to show how we can turn these disparate data streams that are generated from these experiments into high-quality data sets that can then be used in a whole slew of automated modeling approaches. And then finally, we're going to talk about how we can apply the learnings from the generation of these predictive models towards minimizing a late-stage process development effort. So this slide shows a general schematic of the automated platform that we're currently developing. So the first step here on the left, the box on the left there, is to generate data sets which quantitatively relate the effects of process inputs to product outputs. So here, when we say product input, we're talking about things we can control, things like feed concentrations, for instance. Whereas product outputs are attributes that we suspect are important in determining the quality of the medicine. So we can generate these data sets using techniques that are borrowed from a well-developed field known as systems identification, which we'll talk a little bit more about later in the webinar. In the approach, we can generate high-quality data sets in a continuous bioreactor in which we separately manipulate the concentrations of multiple feed components in the reactor. The actual components that we manipulate and the concentrations of the components that we choose is determined through a combination of prior knowledge of the process platform as well as established best practices for these types of experiments. So during these experiments, while we're manipulating these feed concentrations, the reactor is frequently sampled using an automated sampling technology to generate a product quality versus time data. So here it's worth mentioning that the adoption of automated analytical technologies is really critical for being able to collect frequent enough product quality data to be able to relate the effect of each process input to its corresponding effect on the product outputs. Without the appropriate use of automation here, the analytical burden of using this type of approach would be prohibitive, particularly in sort of a lean early phase development shop. So finally, the data sets that are generated from those experiments are curated and turned into predictive models in which we can use data to quantify the effect of each process input has on the measured product quality attributes. So in the end, we essentially end up with a forecast of process behavior that has been constructed using an automated collection of high-frequency data sets. And then depending on the quality of this ultimate prediction, more data can be generated to improve the model prediction if necessary. So kind of the second step here, the two main goals of generating these predictive models is really to both, one, be able to optimize process inputs to achieve the desired product quality attributes, or essentially come up with an optimal feeding profile for across a bioreactor run. The second goal of generating these predictive models is to allow potentially active control of product quality attributes later in development and manufacturing. So in both of these applications, the predictive models that we described in step one are used to generate a forecast of the process outputs, and those forecasts are based on the current process measurements. And so you can think of these models as being very similar to weather forecasts, where in a weather forecast you have a bunch of measurements all across the country of temperatures and pressures and wind speeds, and they use that snapshot plus a history of all the weather leading up until now to essentially try to make a forecast of what's going to happen over the next 7 days. And so you can think of the types of models that we're trying to deploy here as doing a very similar thing. We're making a bunch of measurements of the product quality attributes in the bioreactor, and we're trying to make a forecast of what's going to happen in the future and then act accordingly. This is known as model predictive control. So I mentioned earlier briefly that this data generation platform borrows heavily from a well-established field known as system identification, which has been applied to other industries such as the petrochemical industry and other chemical processes essentially since the 1970s. So system identification in a nutshell uses statistical methods to build mathematical models that describe how a system changes over time. And it does this using data that's actually collected from the process itself. In other industries where system identification has been applied, the benefits of this approach have really been best realized on processes which are relatively complex, and processes where time constraints really just don't allow for individual DOE-style experiments to develop an in-depth fundamentals-based control strategy. So these system identification approaches seek to take and combine sort of the existing expert knowledge of the process fundamentals, and then combine that expertise with a heavily empirical, data-driven approach so that we can make process decisions and develop control schemes quickly and based empirically on the data that we have at hand. So now back to the context of cell culture, using these system identification approaches to generate prediction of cell culture processes has many potential benefits. Firstly, system identification approaches typically do allow for multiple variables to be manipulated simultaneously in the experimental design. And that's the case even for complex systems. And so this would obviously create a potential time savings, particularly for a highly multivariate system like a cell culture process, where we would ideally like to manipulate lots of different variables and see what their individual effects were on the control strategy, the process outputs. Secondly, the actual execution of system identification experiments on a bioreactor is systematic, and that means, from our perspective, that these experiments, the experimental design is actually very easy to automate, which obviously has a time savings associated with it as well. Thirdly, these system identification experiments can be executed in a continuous pseudo-steady state system, which makes directly quantifying the effect of each individual variable on the product outputs easier than a typical traditional DOE-style experimental paradigm where lots of different variables, both the inputs and outputs, are all changing simultaneously. And then finally, and perhaps most importantly actually for system identification approach, is that these types of experiments are designed to yield data sets which are designed with process prediction and active process control in mind. And so this can substantially lower the amount of effort that's required to develop and validate a product quality control strategy later in development. Now I'm going to hand it back over to Jeff, who's going to talk about specifically some of the tools that we've had to develop to enable this approach.
Great, thanks, Brandon. So as Brandon was mentioning just now, there are a number of capabilities, or I should say concepts, that we're leveraging here to advance this integrated platform that we'll be discussing. That was the sort of conceptual section of the webinar. And as we proceed, we'll be talking more in depth, getting into details around hardware and software, as well as experimental design, and actually some results from our internal R&D programs. So the next section of the webinar is really around the technologies we developed in-house in partnerships with our clients and collaborators to really make this integrated platform happen. In particular, we'll be discussing our lab environment, our modular automated sampling technologies we call MAST, and in particular the SP200 valve that's enabling MAST, as well as data handling and curation software we've developed internally to streamline our efforts here and actually make everything go faster. So as Brian was just mentioning, we're really focusing in on system ID-based control process and developing that here at Ben Research. And we started off a number of years back discussing internally around what's needed to develop a system ID-based methodology. And we understood we needed at that point a well-characterized platform that we can actually then apply to, say, early phase R&D work, as well as perhaps to translate to down the line, late stage manufacturing at our clients' shops or internally. One of the things we first developed was an automated system that's using MAST and software. And to use that, we actually developed an operating space, and with the goal of developing an optimized feeding profiles, for instance, as well as we're currently focusing on control strategies and ultimately process understanding. And as I mentioned, we also wanted to focus in on early phase as well as CTM material, perhaps I&D. So the lab environment that we have here at Ben Research that's fairly integrated. We started off with a number of questions or problem statements. We've heard from the industry, our clients, and we are experiencing internally. And so things such as critical quality attributes or CQAs, how we can control those, that was an interesting problem statement we've heard time and time again. Robust data sets, so big data, everyone's getting big data. So how can we utilize big data more efficiently? How can we actually translate that into knowledge and ultimately application? So it's not big data for big data's sake alone. Also, which is extremely relative and pertinent here, is the correct analytical, which is ultimately PAT, integrating PAT into this environment so that it's an automated fashion. We get data out of our PAT in real time, and we can then use that data in a very facile manner. But we're also leveraging here at Ben Research's modeling and applied math skill sets. We have a team here in-house that actually focuses in on development of models, predictive models around big data sets. We're leveraging that as well. So we have essentially, if you go through our lab environment, we have enabling technologies. These are, we call customized solutions, which includes our MAST, liquid sample handling, using a Gilson PAT such as the NOVA. We also have novel solutions like the MAST, and also a cell separation device, which are both hardware solutions. We also have software solutions such as Jubal, which is our applied math and data handling software tool. We also leverage skill sets, engineering, modeling capabilities, as well as biology in-house and chemistry. So what we have here at Ben Research, we've developed this integrated lab, which starts at bioreactors. We've integrated sampling hardware into the bioreactors. We now have also integrated PAT tools, which feed off of the MAST. Data reduction tools, including Jubal, and then application tools, which is our modeling and know-how. And that Venn diagram at the bottom of this slide really shows essentially the overlap of all those disciplines into one cohesive model. This is a rough photograph of our integrated lab environment. We created this lab environment from the ground up. It was a blank slate space. And we said to ourselves we want to have bioreactors integrated with PAT tools using MAST. This has to be automated with electronic data handling to make analysis more easily available for our scientists, both in the lab as well as back in the office for data reduction. An important part of that was modeling and visualization tools. So this is currently we have in this space a number of broadly James reactors. We have the MAST hooked up to our Gilson liquid sampling, liquid handler, as well as the NOVA and LC technologies as well here in this space. First, we'll start off now discussing sampling hardware and integrated PAT. This is the MAST sampling platform I was discussing earlier. MAST is a modular sampling platform that integrates into the biologics manufacturing process. It's able to maintain a sterile environment during sampling and is self-cleaning. It's fully automated with an intuitive user interface. So what it does is MAST integrates upstream and downstream biologics manufacturing processes with analytical tools. It generates real-time observability into manufacturing processes. MAST pushes samples rather than pulls with a vacuum. That's a big differentiator. How it enables in the lab is it generates samples whenever we'd like. It's automated. It interfaces with our bioreactors as well as with analytical tools. And what it essentially does is it enables new ways of thinking about experimental design and data set robustness. So the typical limitations of sampling are sort of taken away using MAST, in that you can generate samples whenever you would like, with the frequency you'd like, and the sample volume that you would like, to get the kind of data out of your reactors that you need to understand your processes. This is the SP200 valve. It's sort of the heart of the MAST program. It enables real-time data, as I was just mentioning. This is the evolution of the SP200, going from the far left to the far right. This SP200, the sampling valve, started off as a collaboration program with a client and evolved into something that's much more than that now. In that, we started off with proof of concept feasibility work all the way through to what we say is a commercial product on the far right here and is available. The new MAST has a simplified and flexible filter integration, has a smaller footprint on the bioreactor head plate to save space. It has a very simple valve seat and pneumatics. It's fully integrated, pneumatic umbilical as opposed to historical versions, and units are available. This is an example application about how we think the MAST and the SP200 are really integral to sort of, in this case, a feedback control scheme. So we think with the bioreactor, the MAST directly and the SP200 directly integrates with the bioreactor, feeds into PAT tools, which then lets us know about quality attributes in real time. We can then take those large data sets, put them into model predictive control, sort of applied math applications. And we can manipulate feeds and feed set points based upon the forecast models that are generated from those large data sets, which then feed back into the bioreactor. This is one very simple way that we're currently thinking about how MAST can be used in the lab setting. So far we've discussed the lab environment, the SP200 and PAT integration. Now we'll move on to data reduction tools. So this is really turning these MAST-generated samples, the PAT-generated data, into real know-how of that bioreactor. One of the main goals here of the program at Ben is to remove that black box from the bioreactor, which means getting to understand in real time what's happening in that bioreactor, regardless of if it's a batch, fed batch, or perfusion-based reactor. And we think this sort of sampling frequency with bright PAT integration can really then generate large data sets. One of the ways that we're actually looking at using this is a software tool we developed in-house we call Jubal. The goal of Jubal was to take real-time quality attribute data, and CQAs could mean galactosylation, sugar residues, it could mean media profiles, and then begin to understand how we can apply that CQA data within the context of process data. As shown on this slide, we have capacitance and ammonia as a function of time. This is day since inoculation. And what you can see is we can plot in real time using the mast feeding samples to our NOVA. We can plot ammonia in real time, which is great because this is actually one key variable and indicator of, say, cellular performance. Other data streams can be integrated into mast, which is what we're currently doing, to understand how these data correlate with each other and also begin to build models off these data sets using Jubal. This is the Jubal home screen and experimental creation. Moving from left to right, we can see that Jubal is not only a data visualization tool, it's also an experimental design tool, whether that's a reactor or a shake flask study. We can then visualize in real time here in the center in blue, visualize data streams in real time coming out of our bioreactors or inputs from shake flask studies and our lab scientists. It's also a historical curation tool in that we can actually look at historical data sets using Jubal so we can compare ongoing runs to historical runs to see how our current bioreactors are performing without having to go back to our desks potentially and looking up Excel spreadsheets. And essentially, this gives us real-time knowledge of our performances currently and historical. Which brings us to the application stage of this story, more or less. As we go from hardware to software to data, now we're talking about how we're actually using and leveraging this platform within a real sort of experimental design situation. And from this standpoint on, Brandon will be discussing an IR&D program where we've looked at galactosylation as a function of galactose inputs and how we're using this integrated lab methodology to streamline and increase the robustness of these sort of studies. And so Brandon and I will hand over to you the slides.
Thanks, Jeff. So in this final section, as Jeff mentioned, we're going to go through a case study where we essentially executed a step response experiments, which are a very simple systems identification experimental methodology. And we did that so that we could quantify the effect of galactose feeding on protein glycosylation. So I'm not sure how diverse of an audience we have on the phone today, but for those that are maybe less familiar with biotherapeutics, glycoforms are essentially sugar chains which are attached to proteins by the host cell as a post-translational modification. And so often the identity and the specific configuration of the sugar groups making up each of these glycoforms, each of these sugar chains, affects the efficacy and the pharmacokinetics of the active protein in vivo. And so this makes the specific identity of the glycoforms that are attached to these proteins an important determiner of the product quality. So glycosylation is often hard to control, actually, because it's an intracellular process. What that means is that it's sensitive to changes in cell physiology. And then cell physiology itself, obviously, the cell is constantly adapting to a number of factors, including changing media and process conditions. So because of the importance of the glycosylation profile as a product quality attribute, combined with the inherent difficulties involved in its control, we chose to demonstrate a process characterization methodology here in the form of a glycosylation case study. So here we're going to demonstrate how this process characterization methodology using a simple system identification experiment can be used to characterize the effect, characterize and quantify actually the effect of feeding galactose on the percent galactosylation of the expressed antibody. So our data collection approach here was targeted at gathering data for characterizing several different aspects of the process behavior. In particular, we wanted to know, firstly, where does the process tend to go when we change a certain particular process input. And so here, for the purposes of this case study, we're calling that the steady state answer, or the steady state data. And this is the most commonly collected type of data from bioreactor experiments, because it really tells you, in general, where is the process going to go over a long period of time, and you can kind of generate these results with relatively little experimental effort. So for these, for this study, we want to not only measure the general process tendencies or the steady states, but we also wanted to know how and when the process arrives at each of those steady states, so that we could use that information to develop a predictive dynamic model of product quality over time. And so here we're going to call that dynamic data. So you can see here the graph on the right. There's sort of you can see at T0 we change the we change the galactose concentration in the reactor, and we see a response change in the percent galactosylation over time there, and you can see that eventually we reach a new steady state. And that would be equivalent to the steady state answer, which is summarized in a graph that looks like the graph on the left there. And so finally, we wanted to measure both of these pieces of data under constraints that would create the best quality data set for predictive modeling purposes. And so for this study, that means we wanted to build an apparatus that could manipulate the important variables, feed variables that affect the output, obviously in this case galactose affecting percent galactosylation. And we also needed to be able to manipulate those variables in the correct dynamic range. So we don't want to be testing variables outside of a range where we would either have a deleterious effect on the growth or metabolism of the actual cells themselves, and we wouldn't want to test in a range that's so low that we don't see any response in the protein quality. And then finally, and probably most importantly, we wanted an apparatus where we would be able to separate out the individual variables and manipulate them individually if desired. So to capture this type of information for this case study, we decided to run a very simple systems identification experiment where we put the cell culture into a pseudo-steady state using a perfusion apparatus. And so this allowed us to manipulate our chosen process input, in this case the galactose concentration, separately from all the other feed variables, all the other feed components. So we chose to excite the dynamics in this case by providing step inputs in galactose concentration to the system. So you can see the graph that's on the screen right now. Essentially what we're doing is we're executing an experiment where we're in a staircase type fashion increasing the galactose concentration in the reactor, and then essentially using our automated analytical suite to measure the effect that has on the percent galactosylation, which is the output in this case, over time. And so that's shown on the lower graph there in the green. And so the final step to this obviously is all of that time course data is collected and curated electronically and then further analyzed. So that we can pull out the key steady state and dynamic characteristics of percent galactosylation of the antibody. So shown here is the type of data that you get out of that kind of a study. So firstly, we can observe kind of what is expected in this graph on the lower left here in terms of galactose concentration over time for this study. So the axes on this graph here briefly are the orange dotted line is the galactose feed concentration. That's the concentration of galactose that's coming in with the perfusion feed in this case. And then the blue trace is essentially the measured galactose concentration in the actual reactor over time as measured using an autosampler. So you can see that as expected, when we first step up to a new galactose concentration, the measured concentration in the reactor sort of equals the concentration that's in the feed. But then after a while, the cells are sort of consuming down that galactose, and they reach a steady state value where the galactose concentration in the reactor is lower than what's in the feed, essentially. So that's due to the cells consuming galactose at some steady state rate. The other piece of information, obviously, that we wanted to get out of this kind of a study is what is then the corresponding effect on the percent galactosylation versus time. And so that's shown on the graph on the lower right there. And so you can see that for each step change, with the exception of the kind of initial steady state equilibration, which is kind of from day 5 to day 20 in this particular experiment, where that's just the culture adapting to the perfusion operating conditions with no galactose added. You can see that for the first two step changes to 10 and 100 micromolar, really don't see a significant effect in increasing percent galactosylation. However, when we go up to 1 millimolar and 10 millimolar concentrations, you can see that we do see a dynamic response, and the percent galactosylation reaches a new steady state value, which is higher than the previous condition. So we were able to take that data now and pull a couple of key pieces of information out of it. From the galactose data, we can come up with a steady state galactose consumption rate, and that's just kind of governed by the offset between the galactose concentration that we're feeding and the galactose concentration in the reactor. And likewise, for the galactosylation or the glycosylation data, we can pull out a steady state value from that too. So the glycosylation obviously reaches a new steady state eventually when they're subjected to a different galactose concentration. But interestingly, we can also pull another piece of information out of the galactosylation data set, and that's a rate piece of information. So we can also ask ourselves, not only where does it go when I subject that culture to a certain galactose concentration, but also how long is it going to take it to get to that new steady state. And so these are key pieces of information when you're trying to enact a control scheme where you're trying to control things through time. The pieces of information that we're going to want to know in order to make a forecast are not only where's the process tending to go, but also how long is it going to take to get there. So if we just kind of summarize the three key results that we get from these data sets, you can see that we can learn a lot about both the fundamentals of how galactose is affecting galactosylation. For instance, galactosylation might actually be limited by galactose uptake rate, for instance. It seems to exhibit Michaelis-Menten kinetics, which we were able to pull out very easily in this experiment. And then, as I mentioned before, it also gives us this dynamic data. And we can see here that it actually approximates first-order dynamics, which is actually very, very conducive to predictive modeling. And so kind of our next steps here in the process is to take this fundamentals-based data combined with increasing the robustness of our data set, so essentially getting more N's on our step inputs, and actually go through the exercise of developing that predictive model for percent galactosylation over time. And then obviously we want to continue to automate and streamline these real-time technologies as we kind of have more and more success with the approach. Now I'll turn it back over to Jeff, who will wrap it up.
Thanks, Brandon. So more or less in summary at this point, It's widely believed, and we believe, that biologics process development is on the critical path. Essentially what that means is, for us and for a number of our collaborators, that if we increase the robustness and quality of the upstream processes during early process development, we can, in fact, as shown in the bottom of this slide, shorten and streamline biologic development into the clinic, as well as being able to increase the amount of candidates we have going into this pipeline. So at this point, we're sitting back and asking the questions, what have we done? What are we trying to do? We believe we've created or begun to create a system that can be used to develop process control systems. This methodology is customized, can be customized based on quality attributes, individual processes, and overall parameters of interest. What is different about this approach? A number of things, but in particular, it leverages the ability to produce high-density data sets using math and SP200. We're using real-time integrated PAT, as well as in-house capabilities such as the engineering, biologics, in particular, the applied math and modeling capabilities here at Bend Research, which we think are really value-added drivers for this approach here at Bend. So thank you for the time, and now we will be happy to answer some questions. Back to you, Sam.
Thank you very much. So yeah, it's time for our Q&A session. I'll give another quick reminder of how you send those in. You either click on the Q&A widget at the bottom of your screen or type them into the white box at the top left-hand corner and press submit. We've already got a few in, so I'll go straight into the questions I've already got. And the first one I'm going to go to is, has this strategy been able to rapidly detect microbial contamination in bioreactors when it has occurred?
Brandon,
Jeff, I guess I could do.
Yeah.
Sure, yeah. I think, there's definitely an opportunity there. That's not something that we've, gone after testing explicitly with the system. However, having that really high sampling rate, having a sample every few hours, really would aid in being able to detect things like that earlier, depending on obviously when the contamination event actually occurred. I guess a side note there is these reactors are actually being the cell density in each of these steady states is being controlled using capacitance, and there actually is a potential for that tool to be applied, which is actually an online technology as well for early microbial contamination. But there's also an opportunity, I would say again for using math combined with like a NovaFlex or something like that, where you can really particularly if you're looking for changes in metabolites, you can kind of detect those really early on, and provide the analytical instrument has the sensitivity to detect it.
Okay, thank you very much. Next question I've got is, if the intercellular process is difficult to manage, how is contamination, cross-contamination, and cleaning assured between manufacturing runs? Something residue-free, quick, and easy to use, maybe?
So I guess I'll begin to answer this question by saying the SP200 can be autoclaved. So essentially, the bioreactor is built, and the SP200 is placed on top of the bioreactor and autoclaved, and then hooked up to the PAT in a sterile fashion in the lab setting. It's sort of how we manage sterility and cleanliness. As well, we have the SP200 and the SP100, which is a larger sampling valve, and we can use steam or liquid sanitant to clean those valves based upon our client's specific sanitant of interest. That's something that's actually a discussion point with our clients, which is what sort of system do you envision this being used on? What sort of sanitant is used in your setting, and how do you want to maintain sort of cleanliness and setup of your bioreactors? That's really how we manage sterility and sort of basic bioreactor hygiene using our physical setup with our hardware tools.
Jeff, I guess one thing to add to that too is that all of the PAT technologies that we've presented today can be adapted to a disposables application, which obviously has benefits there as well.
Yeah.
Okay, thank you. We've got a few short questions now. First one is, how frequently can you get the data per minute?
So I can answer that from a mass perspective. That really depends on how thorough you want to be about assuring sterility between mass samples. Typically, in the experiments that we're presenting here, the dynamics are sufficiently slow so that one sample about every 4 hours for mammalian cells is really plenty of data to capture all the dynamics that are happening. The maximum, I would say, that people use a mass for in cell culture traditionally is really about 1 sample per hour. Although I think you could probably get it down to 1 sample per every half hour. I think we've done that before. But I'd say that would probably be the upper limit, since you have to sanitize between each individual sample.
That's right. I think Brandon's got a good point there, which is biologic dynamics are usually slower than that. So our sampling rate is faster than what you often see from dynamic standpoints out of a bioreactor, which is, I think, extremely valuable in this setting.
Okay, thank you. Next question is, how many reactors can you run this side by side?
I guess,
yeah, so,
Brandon, I guess I'm thinking here is how many reactors we hook up to a mast and have fed to our, say, a NOVA, which I guess gets back to, so the geometry is one SP200 to one reactor, and then we can then shuttle samples through the traffic cop, the navigator, into, say, a NOVA or a Gilson sample collection tool. And one thing, I guess, I don't know if we made this very clear in the presentation, is that the mast system is very modular. We have a basic mast system that's available, but at the same time, if our clients want more than a basic system, they want to make it modular for their PAT tools that they have in-house. That's actually a valuable discussion point to have with us, because this thing is entirely modulatable for our clients' lab setting.
Thank you. Next one is, how long is the development time?
Yeah, I think the thing to say there is that it really depends on how fast the dynamics are that you're measuring. So in the case of this percent galactosylation, we were seeing settling times that were kind of on the order of days. So that means that essentially you're looking at several days per condition, essentially, for this study. However, as I mentioned earlier in the talk, one of the things that we're working on, and one of the things that systems identification is really good at, is expanding that to being able to manipulate multiple variables simultaneously. And so that's where we really see the time savings over kind of what's being done currently, is that we see a big time opportunity to be able to manipulate multiple variables simultaneously and then pull predictive models out of that data.
Okay, thank you. Got time for one more question before we close, and that is, how frequently do you generate samples using MAST, and how are you using those kinds of data?
Yeah, I would say that this gets back to the quality attributes that we're trying to quantify with our system, which gets back to what's important for our clients from a process perspective, which also obviously gets back to the PAT that we're going to tie into the system. So the frequency of sampling, the type of data we're generating, And sort of the processes, whether that's batch fed, batch perfusion, really gets back to the problem statement our clients have. And that's sort of how we take a very high-level look at the problem statement that they're bringing to the table. And then we can design a customized solution around that problem statement, leveraging sort of all these hardware tools and our current thinking around data handling and modeling to bear. So really, it's a problem statement-driven point there, and we can customize that discussion and sort of what we want to do going forward based upon that. But really, it's around CQAs, whether that's a process CQA or a protein CQA. What are we trying to optimize, and how can we go about doing that? Brandon, anything you wanted to add?
No, Jeff, I think that sums it up great.
Yeah.
Okay,
Great.
Thank you very much. So that brings us to the end of the Q&A session and the end of the webinar. Just leaves me to thank Jeff and Brandon for what was a great presentation, and Ben Research for sponsoring this session. So all the attendees, you'll receive an email shortly in a couple of hours telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. Once the webinar ends in just a moment, you'll see a survey appear in its place, and we'd appreciate if you took the time to answer the questions in there. We look forward to sharing further webinars with you, so please keep an eye on the website I just mentioned. Follow us on Twitter, which is @BRWebinars, for daily updates, and join our LinkedIn group, Business Review Webinars. Apart from that, thank you once again, and hope you all have a nice day.